In this video, what we're going to do is to review the chemical structures of the macronutrients whose digestion and absorption we're talking about this week. What I want you to come away with is when you're reading this text about absorption and digestion, and particularly with the metabolism, I want you to have a picture in your mind of what all of those words mean. So let's get started. All right, so I want to start with carbohydrates. Remember that carbohydrates come to us as ring structures for the most part, except for glycerol. They have oxygens, hydroxyl groups, hanging off of those rings. So the basic structure of all carbohydrates are these things that we call monosaccharides. The three that we are going to find the most is glucose, galactose, and fructose. That's what's in our foods. There's also a molecule called glycerol that is attached to the fatty acids to form fats. That happens to be a carbohydrate, and our body can use it to make glucose in a process that we're going to talk about later. But for right now, let's just talk about carbs. All right, so these three, if you can ingest just any one of these are what's directly absorbed through the intestinal wall. But our food does not come in this form. You cannot go to the supermarket and buy pure glucose, galactose, or fructose. Instead, the way that these usually come to us is as either disaccharides or as polysaccharides. So the three most common disaccharides are sucrose, lactose, and maltose. And these are formed from the combination of two of those monosaccharide subunits that we saw. So sucrose is a fructose hooked up to a glucose. Lactose is a glucose hooked up to a galactose, and maltose is two glucose units hooked together. Before these can be absorbed, they have to be digested back to their monosaccharide structures. So after digestion, sucrose becomes a glucose and a fructose, and those two can be absorbed as is. Starch. The way that a lot of our carbohydrates comes into our body is from that storage form that fruits and vegetables make, that plants make, which is as starch. Starch comes in two forms, amylopectin and amylose, and the form has some effect on how quickly they're digested, but from the point of view of digestion and absorption, the end result is that these chains, which are polyglucose, nothing else, no galactose, no fructose, they are broken down through the process of digestion by enzymes into glucose, lots of it. And then only after they are broken down into glucose can they be absorbed. Glycogen is another form that we are going to be encountering. This doesn't come from plants. We make it ourselves as our storage form of glucose. And it is similar to starch in that it is just a bunch of glucoses strung together. But there's some details about the linkages and the way that the enzymes that make them put it together. But it doesn't matter. What our body has to do before we can use the glucose that's in here is break those chains apart and turn it into glucose before we can absorb it. All right, so let's turn now to proteins. I want to remind you that proteins are made out of amino acids. Here's a generic amino acid. The basic chemical formula for all amino acids is there's an amino group on the end, there's a carboxylic acid at the end, that's how they get their name, amino acids, and then there's a central carbon to which is attached another group. this is a placeholder. It's called the R group. And what that means is that what's up here can be any of 20 different things. Here are two examples of those R groups. For this particular amino acid, which is glycine, the R group is just a hydrogen. That's the simplest amino acid. But this is one of the more complicated ones. This is phenylalanine, and you can see that the group that is up here attached to that central carbon is this big old phenyl group. It's a big hairy ring structure. Any one of these can be absorbed as is without further modification. So during digestion, the goal is to get the proteins that we take in through our diet down to this form so that they can be absorbed across the intestinal wall. But this is how the proteins that we eat come into our digestive system. They don't come as amino acids. So to get them from amino acids, you have to reverse the process by which they're synthesized. So let's talk about how amino acids get to be this. So first of all, you have 20 different amino acids. Meaning there are 20 different types, but to make a protein, typically you need about 100 to 1,000 amino acids. It may be 5 tryptophans, 50 glycines, 20 alanines, whatever. The combination is different for each protein, and that is dictated, of course, by the sequence of the RNA. And you guys talked at some point about how proteins are made, how genes get turned into proteins. So, but from those 20 amino acids, through a process called translation, the 20 amino acids get put into a chain where they get joined together. The N group, amino group, joins up with the carboxylic acid group into what's called a peptide bond. So these 20 different amino acids are put into a chain. And this chain is what we call a protein. Another term for this is polypeptide. So usually the reason that we would use the word polypeptide is that we want to emphasize how many amino acids there are. But polypeptide and protein means the same thing. So after these chains get put together, unlike the carbohydrates, what makes proteins work is actually their three-dimensional structure. This here, if it's strung out like that, does nothing for you. So to go from a polypeptide chain to a three-dimensional intact active protein, it has to fold. And the folding instructions actually come from the amino acids themselves. So spontaneously, they will fold up into a three-dimensional shape. And that's how it is that they come into our diet, into our foods. So this is how it gets into our body. We eat them and it goes through our mouth. Nothing happens to proteins in our mouth. There's no digestion that takes place there. But once it gets into the stomach, that's where the digestion of proteins begin. The first thing that happens is that the acid that is in the stomach denatures the protein. Denaturation means to reverse that folding process. Strip them, turn it down into a chain. But no peptide bonds are broken here. So after the acid denatures the protein, the peptide bonds that are in here become accessible to an enzyme called pepsin, which breaks down that chain into peptides. peptides, how many of them? Sometimes 2, sometimes 1, sometimes 50, sometimes 100. What pepsin does is it looks for a particular combination of amino acids and breaks between them. So how big the peptides are depends on how many of those sequences that it finds in the chain and how far apart they are spaced. Nevertheless, the digestion that pepsin does is not enough for it to be absorbed. From pepsin digestion alone, what you will get is a mixture of peptides of different lengths, and this cannot be absorbed. So after pepsin digestion into chunks, the next step takes place in the pancreas. In the small intestine. The pancreas dumps enzymes into the small intestine that takes the peptide chains here and hammers away at them until they are reduced to mono, di, or tripeptides, no more. And in this form, with 1, 2, or 3 amino acids chained together, the intestine is able to absorb amino acids at that level. One point I wanted to make here is that if the proteins are not denatured in the stomach, then none of this takes place. And what will happen is the proteins will just pass through the digestive system and out of the body. All right, let's turn now to fats. The form of lipids that we can use to make energy are the triglycerides. There are other fats that we consume in our diet, phospholipids, cholesterol. Some of them have functions in the body, but these are the only ones that we can get energy from. Triglycerides are formed from 2 types of molecules. One type is this glycerol. Remember I was talking earlier about how glycerol is a carbohydrate? It's not in a ring structure. Instead, it's just a linear structure. It's too small to form a ring, but here's those telltale hydroxyl groups, and that's what makes it a carbohydrate. So fats, or triglycerides, are connected to these structures here, which are called fatty acids. Fatty acids are long hydrocarbon chains, but they have a carboxylic acid group on one end that can join to this hydroxyl group to form a triglyceride. And this is what we call fats. They're called triglycerides because each one of these hydroxyl groups here is hooked up to a fatty acid. These are fatty acids. Before fats can be digested, they need to be emulsified. Do you remember we were talking about proteins? Proteins cannot be digested unless they're denatured. Same thing goes for fats. Before they can be digested, they have to be emulsified. Otherwise, they just pass through the digestive system. So imagine this is what fat looks like when you eat it. Think of a big chunk of that stuff that's hanging off the edge of your steak. So in the small intestine, bile is secreted into the small intestine from the gallbladder, and that bile will interact with the fats and break them up into goblets. Once it's broken up like this, the triglyceride bonds are susceptible to digestion. It cannot be digested unless it's emulsified. So once they are emulsified, then there are some enzymes called lipases which will break down the fats. What they do is they break them down into glycerol and fatty acids. The glycerol and the fatty acids are what are absorbed. You can also absorb a monoglyceride, a single fatty acid connected to glycerol, but you cannot absorb diglycerides or triglycerides. So there are small amounts of lipases in the mouth and in the stomach, but these lipases really don't do much. They're mainly there to enhance the amount of fat that you can absorb, but we could do without them. They don't do that much for us. The bulk of the digestion takes place in the small intestine. And here it is that the lipases break off the fatty acids from the glycerol to form glycerol, monoglycerides, and free fatty acids. And those are the forms that can be absorbed.